Okay, so continuing on with our series of lectures, this is going to be carbohydrates and glycoconjugates of cell surfaces. So we're going to start with this fun little piece of work called The Discovery of Honey, and we're going to talk a lot more about carbohydrate metabolism next semester, but I think it's particularly relevant to just put to the forefront of our minds that mammals love sugars. For very, very good reasons. One, because of the energy, first and foremost, but two, one of the things we're going to talk about more in this chapter and introduce the concept of is the idea that carbohydrates act as signaling compounds, act as structural compounds, and modify proteins, altering their structure, function, and regulation, and are also key for immune response. So on top of energetics, there's a lot of really important facets to carbohydrate chemistry that we're going to scratch the surface of in this lecture. If I can figure out how to advance everything. Here we go. Oh, got a little bit out of control there. So here's the general outline for the day. We're going to talk about nomenclature of carbohydrates, particularly stereochemistry. We're going to talk about the different categories, including monosaccharides, oligosaccharides, and polysaccharides. And then we're going to introduce the idea of glycoproteins, their structure, function in the cell. We'll talk a little bit about proteoglycans and their roles. And we'll also talk about the structural roles of the classic structural carbohydrates. So let's do a quick review. Everyone here's had organic chemistry, which is great, and hopefully everyone remembers what an aldehyde and a ketone are. The two general classes of carbohydrates, monomeric carbohydrates, are aldoses and ketoses. And aldoses are aldehyde-containing carbohydrates, whereas ketoses are ketone-containing carbohydrates. And these different classes of carbohydrates can have different numbers of carbons in their backbone. So we can have trioses, tetroses, etc. Based on the number, and that would have 3 carbons in the chain, 4 carbons in the chain, 5 carbons, etc. So when we talk about stereochemistry of carbohydrates, aldoses with 3 or more carbons in their chain and ketoses which have 4 or more carbons are going to be chiral compounds because of the inherent asymmetry in their structure. And when we're talking about the conventions of carbohydrates, we don't use R and S because biochemists established a convention a long time ago. And we're not going to use small d, little l. Remember, those, when you look at dl, from the organic chemistry perspective, typically that's going to be meaning dextrorotary, levorotary, where it has to deal with the handedness of the way light is bent when it's applied to these pure compounds. Capital D, capital L has nothing to do with the rotation of plane polarized light. It has to do with referencing the stereocenter of interest back to D and L glyceraldehyde, which we talked about with amino acids. So just like amino acids having glyceraldehyde as a stereo analog, we're going to do the same thing with carbohydrates because of this very old biochemistry convention. And it is what it is, so we're just going to have to get comfortable with the fact that it is not the most elegant way to name carbohydrates, but it's the way they're named. So getting on board. So for the next couple of slides, all of the sugars are going to be the D variation of the sugar, and that's because D sugar is overwhelmingly predominant in nature. Oh, let's see here. Was there something else? No. Okay. So let's take a look at structure of monosaccharides first, and we'll just build from there. So these are your simplest aldose and simplest ketose coming up. We've got glyceraldehyde, which is an aldose because it's an aldehyde, and we have 3 carbons in the chain. We've got, let's see here. Let me grab a let's see if I can change my, nope, I can't. Okay, well, we'll just use this cursor because I've committed to this cursor, it appears, unfortunately. So we have carbon chain, 1, 2, 3 carbons in the backbone, and we can have an L or D isomer, and that isomer is going to be determined based on the stereocenter of the highest numbered asymmetric carbon, which in this case is the second carbon for both of these chains. We also have a different view of it. If we were to look at it from above, we've got our L-glyceraldehyde here, and we've got our D-glyceraldehyde here, and you can see the difference in handedness if you were to assign the different functions coming off of the central carbon 1 through 4. So let's blow up the aldoses a little bit. We started with D-glyceraldehyde in this, and we need to keep in mind that for every D version of these carbohydrates, if you take the mirror image of the entire structure, you'll end up with the L variation of it. So you could draw any of these L versions simply by doing the stereo or the mirror image of that particular structure. But for all of these cases, we're going to be dealing with the D variations. So we've got our D-glyceraldehyde here, and it's, like I said, D because of the orientation of the hydroxyl group off of the highest asymmetric carbon. So in this case, it's the second carbon. And then if we add another carbon to the chain, and we get 4 carbons here and 4 carbons here, we have the ability to add another stereocenter based on the orientation of the hydroxyl group on that new carbon. So the new carbon being highlighted in yellow, in this case, the hydroxyl group is off to the right, and in this case, it's off to the left. And that's going to make 2 new compounds versus 1 new compound. This will be D-erythrose, and then we'll have D-threose over on this side. And those will be our aldotetroses. So we've got aldotetrose being aldehyde-based, CHO, CHO, and tetrose, 4 carbons per chain. Then if we just elaborate off of D-erythrose out of convenience, we can move yet another new carbon in. And so the 2 carbon is going to be the new carbon with a new chiral center, where we can have a hydroxyl group on the right side or a hydroxyl group on the left side. And this is going to create 2 new carbohydrates, D-ribose and D-arabinose. If we do the same thing for the threose, we'll get D-xylose and D-lysose. Notice that as we add these new chiral centers, these new carbons, that the box in pink keeps getting moved down the notch. It keeps getting a higher number ascribed to it. And so if we were to ask, why is this a D sugar? The reason it's a D sugar is because the orientation of the hydroxyl on the highest numbered asymmetric carbon is to the right. So we can do this one more time with ribose. We can add yet another new carbon. This will give us yet another new chiral center, and this will give us allose and altrose. Now, if we expand it off of arabinose, you'll see something a little bit more familiar. We've got our D-glucose and D-mannose. Both of these are really common, particularly the D-glucose. So what I tell people when they finish this chapter, you're going to have a lot of carbohydrates in your head, and moving into next semester, the 2 carbohydrates you have to have down stone cold are your glucose and your fructose. You say, Dr. McGill, there's no fructose here. Aha, that's because it is not an aldose. So let's move on to aldoses. Excuse me, to fructoses. Fructose is going to be in the ketose category because it, as a straight chain carbohydrate, has a ketone functionality. And because it's a ketone, that carbonyl group has to be on the second carbon. It's not on the first carbon like on the aldehyde, aldoses. And because of this, you have to add an extra carbon to the chain before you get a stereocenter because of the lack of a fourth group around the central carbon here. So our most basic ketose is our ketotriose, which is dihydroxyacetone. And we're going to see a lot of dihydroxyacetone as a metabolic compound in metabolism, fundamental metabolism next semester. But it's not going to get that much attention from us in this chapter. If we add another carbon to this chain, we're going to get our ketotetrose, and that's going to establish our first asymmetric carbon. And this will be deerythrulose. So once again, because the hydroxyl group is off to the right side, the way it's drawn here, this is going to be deerythrulose. If we put a mirror right here, and we did a perfect mirror image of this entire compound, not selectively only inverting carbon 3, but inverting every single position, what you'd get is L-erythrulose. So moving on, we're going to do the exact same thing we did previously, where we add new asymmetric carbons, where the hydroxyl group is off to the right here, off to the left here. This would be our ketopentoses, and we'll get our D-ribulose and D-xylulose. And if we do it one more time, we're going to get our fructose down here, and some much more obscure carbohydrates that I'm not going to be quizzing you on because this is all, these are, like I said, obscure. So fructose, very important. Glucose, very important. Some of these others like erythrulose, ribulose, you're going to see those periodically. We're going to see a lot of dihydroxyacetone later next semester, but not in this chapter. So if you were to encounter a question regarding stereochemistry of carbohydrates, and in classic multiple choice exams which deal with the biochemistry of carbohydrates, there's almost always going to be some kind of question regarding stereochemistry. Remember that stereoisomers, which are non-superimposable mirror images of each other, are enantiomers. So if you take a mirror image and you can no longer superimpose that mirror image through any three-dimensional rotation, that's going to be an enantiomer. A diastereomer is when you have a pair of isomers which have opposite configurations at one or more chiral centers but are not mirror images. So in that case, you haven't inverted every single invertible position. You've only inverted a few of them. And that's going to give you a diastereomer. So what we're looking at in these previous slides are examples of diastereomers in any two rows. So let me hop back here. Erythrulose and threose will be diastereomers because every carbon, which is chiral, is the same except for the second carbon in the chain here and here on the yellow positions. Now, another term that you see occasionally is the term epimer. And an epimer is whenever you have a difference in configuration at only a single chiral center. And technically, these are diastereomers. However, because they have only a single positional change, epimer is also used. So once again, if we went back, if we were to compare erythrose and threose, these would be epimers of one another. And moving along. Now, enantiomers, I've mentioned, are perfect and complete mirror images, which are non-superimposable. So in this case, if we have D-fructose drawn with the hydroxyl group at the third carbon over to the left, and the hydroxyl groups on the fourth and fifth carbon to the right, when we put a magic mirror right down the middle here, oh, clicking away, getting excited. If we had a magic mirror down the middle, near side steers on near side, far sides on the orange go over to the far side over here. Non-chiral positions don't matter. This would be an example of an enantiomer, and this would be what differentiates a D-fructose from an L-fructose. So if you were given a picture of a D-fructose and said, hey, draw an L-fructose, you just throw up that mirror mechanism that you learned in OChem and draw and invert every stereocenter, and you would have the L variation. And an antinomeric pair by way of having done that. So let's see here. Many of you at this point have been thinking, all of these structures are linear structures, but I'm pretty sure in previous courses I've seen pictures of carbohydrates, and both glucose and fructose and a lot of these carbohydrate structures were drawn as cyclic structures. And you'd be correct, because these linear structures readily biologically cyclize as part of equilibrium processes, and the cyclic structures are predominant. So, getting click happy. Sorry, everybody. So the cyclic monosaccharides form readily in nature, and like I said, predominate. So glucose, which is your classic aldose, can cyclize via forming a hemiacetal internally, and fructose, your basic ketose, can readily cyclize forming a hemiketal. And I'm not going to quiz you on say, hey, draw a hemiacetal, but the mechanism of hemiacetal and hemiketal formations are consistent with what we're about to do mechanistically. So when both the hemiacetal and hemiketal form, the carbonyl carbon atom, both at the aldose, at the aldehyde, and at the ketone, becomes a new asymmetric center. And that new asymmetric center has the ability to, well, I'm getting a little bit ahead of myself, but it's called the anomeric carbon. And the anomeric carbon, if you were doing NMR of carbohydrates, has unique carbon shift, and it has a little bit different chemistry because of its ability to ring open again reversibly. So whenever you have an isomer where the stereochemistry varies at the anomeric carbon, the two isomers are considered anomers of one another. Now, more special terminology. Once again, if you're taking a standardized test, you're going to have to know these. The cyclic form of glucose is called a pyranose, and the cyclic form of fructose is called a furanose, and that's because of their likeness to classic ring structures of a pyran and furan ring. So, in this case, right down here, this is what a pyran ring looks like, and structurally, people who are identifying the structures of cyclic glucose said, man, this looks a whole lot like pyran, even though this has some pi bonds, and it's not exactly the same, but it's a six-membered ring which has an internal oxygen. So humor the classic biochemists a little bit. And the formation of the hemiacetal is when you have a lone pair. It would be nice if they had the lone pairs drawn in here showing you your nucleophilic attack, but they don't. But we've got a lone pair hanging out on this oxygen, actually two, and one of them is going to execute a nucleophilic attack where those electrons push to this carbon. That carbon, remember, is electron poor because of being doubly bound to this oxygen as part of the carbonyl, and so this allows for a nucleophilic attack. Those electrons come in here, they attack this carbon. That's going to take these pi electrons, push those electrons up to this oxygen. Those extra electrons are going to perform a subsequent nucleophilic attack and pick up a extra hydrogen, forming a new hydroxyl group. So this is not the most elegantly drawn mechanism, but if you picked up your old OChem text or did a quick Google, you could flesh it out readily. So this is the formation of a hemiacetal, and we have the components of this alcohol and this aldehyde present here. We have our aldehyde, so we can do this electron poor carbon, and then we have all these hydroxyl groups hanging out. And so the question is which one of these hydroxyl groups going to do the attack? And the one that does the attack most commonly, I'm putting that in as a caveat, but we're going to focus on it first, is the one attached to the 5-carbon, because when the 5-carbon hydroxyl group does a nucleophilic attack on this 1-carbon aldehyde, you get a 6-membered ring, which is a particularly stable ring conformation for all the reasons that you learned in OChem, mostly dealing with bond angles. So the lone pairs here on this hydroxyl are going to attack this carbon, and this oxygen here is going to become a new hydroxyl group. So let's take a look at all the ways this can be drawn and visualized. Here we have our D-glucose, nice and linear, drawn classically. And if we just take it and we take this one carbon and it were to fall over to the right, bang, all the way down. Did I just click? Yeah, I did. Clicking all over the place. What you'd see is the 2 carbon hydroxyl group is down, the 3 carbon hydroxyl group is up, 4 carbon hydroxyl group is down, 5 down, etc. And, 5 would be out because it's going to be performing a nucleophilic attack. So if you were asked to bend this structure, what you do is you just tilt it over to its side with the top carbon teetering over to the right, and then you could wrap it around in a circle, and that could justify this 3-dimensional structure that we're seeing. This also justifies the proximity for the formation of the hemiacetal. Now, remember we were talking about the formation of this new anomeric carbon, this new chiral center at the 1 carbon. And so this carbon, which is housing the aldehyde, undergoes it accepts a nucleophilic attack. This oxygen gets reduced to an alcohol. And now, because this was achiral, it is now going to be chiral because we have 4 different groups around this carbon. This had freedom of rotation. It no longer has freedom of rotation because we're now in a ring. So the fate of this hydroxyl group, whether it's down, like we see here, or up, can happen. Either circumstance can happen. They don't happen equally because of stability reasons. And we'll get into that in a second. But that gives us 2 options. We have the alpha variation, where in the case of D-carbohydrates, drawn with Haworth projection or Fisher projections like this, it's down. So alpha is always down on a D carbohydrate, and beta is always up on a D carbohydrate. Now, that reverses. If these were L carbohydrates, alpha would be up and B would be down. Beta would be down. That's just the easy variation. And we're going to talk about the more correct variation here in a second. But I just want to go one more thing. If you want to draw, so this is your Fisher projection of your non-cyclized, your linear carbohydrate. If you were asked to draw a Fisher projection specifically versus the Haworth projection, which you're going to see more commonly, but if you wanted to draw the Fisher projection, you would just take your Fisher projection that you see over here on the left, and you would take the hydroxyl group, kick off that hydrogen, drag the oxygen all the way up here over to the middle, and then have that bond go all the way up and over to the 1 carbon. And that's what you're seeing here, is you're seeing that oxygen from the 5-hydroxyl moving all the way up and then linking your 6-membered ring, and then your aldehyde oxygen is going to get kicked over to the, in this case, top side, because it's up top. And if this were to tilt over clockwise direction, the 1 carbon hydroxyl anomeric hydroxyl is up, making it beta because it's a D-glucopyranose. And here we have the exact same sequence of thought for the cyclization of fructose, which is hemiketal. And the difference between the hemiacetal and hemiketal is that instead of attacking a aldehyde, we're attacking a ketone. The mechanism is identical. Nicely, they draw one of the 2 lone pairs in this one. And so you can see the nucleophilic attack where the lone pair comes in behind the electron-poor carbon, attacking the carbon successfully, pushing these weaker pi electrons to the oxygen, which can host them a little bit because it is able to do so because of its electronegativity. And then because that's not the most stable place for those electrons to be, it's facilitated that those electrons then reach out and subsequently grab another hydrogen, forming a alcohol group, or in this case a hemiketal. So the exact same thing takes place here, where we have a plethora of hydroxyl groups which could potentially attack the 2-carbon. However, because we're going to end up with a 5-membered ring in this case, it's still the 5-carbon. So the 5-carbon is going to be doing the attack. So lone pairs on the 5-carbon hydroxyl are going to attack the 2-carbon ketone, forming a 5-membered ring. And this is why these are called furanoses, because they resemble the 5-membered ring of a furan, which has that internal oxygen. And everything else about the rationale that we discussed in the previous slide applies here, including drawing a fissure of the cyclic form, as well as what distinguishes something alpha from beta. So, remember, isomers of these monosaccharides that differ by a single position are called anomers. And when we form these new cyclic compounds, we are inherently opening up a window for the formation of a pair of anomers. I introduced the simple rule, where for D sugars, alpha if the anomeric carbon is down and beta is up. For L, it's reversed, as customarily drawn for Hollow projections, that is. However, the classic definition is when the hydroxyl group of the anomeric carbon is on the same side of the Fisher projection as the oxygen at the highest numbered asymmetric carbon. That is considered alpha. So let's walk through that. So let's do alpha D-glucose. So in the case of alpha D-glucose, or alpha D-glucopyranose, if I was to look at the Fisher projection here, the hydroxyl group on the 1-carbon is over on the right side. The highest numbered, going down 1, 2, 3, 4, 5, 6, highest numbered asymmetric is going to be, which has sidedness, is going to be over here. See here. 4-carbon, same side. So 1, same side, makes it alpha. Opposite side, beta. But that really is so much more complex than just remembering alpha down, beta up for all D. And if you're looking at L, just reverse the rule. I would really go with the simple version of the rule in this case. Now, I had a little caveat when I was discussing the cyclization of glucose into forming predominantly 6-membered ring, because the truth is that you can also form a furanose with glucose. When the 4-carbon does an attack on the aldehyde, you can get a 5-membered ring, but that is not as structurally sound and doesn't occur remotely as often as the 6-membered ring. So yeah, this is a bit of trivia, but if you're asked to draw the cyclic form of glucose and it doesn't seem like a loaded question, they're probably talking about the pyranose form, almost assuredly. Also, remember that all of these are going to form chair conformations, which can ring flip by going from a chair conformation to the boat conformation and then to the opposite chair conformation. And whenever you do this ring flip, you're going to be moving things from axial to equatorial positions relatively. And because we have semi-bulky groups accompanied by non-bulky groups being our protons, you're going to have differences in favored equilibria. So in this case, what we're looking at in this picture, 7.2b, are the two possible chair conformations for beta-D-glucose. Notice that beta-D-glucose has, in this position over on the left side of this equilibrium sign, hydroxyl groups, all of the hydroxyl groups and the carbon, the 6th carbon and its hydroxyl group are all in equatorial positions. If we look over to the right side of this, every single hydroxyl and the 6th carbon with the hydroxyl are in axial positions. And so because of this, we know overwhelmingly that the version over on the left is highly favored in equilibrium. So if you were to ask to draw the favored version or the biologically predominant version of beta-D-glucose, you'd be drawing the one on the left. Even though the one on the right can exist, it overwhelmingly is not favored compared to the one on the left. And if you're drawing carbohydrate polymers, justifying the stability of cellulose or glycogen, you're going to want to be leaning on the stability of the predominant form of beta-D-glucose. So, to make things much more interesting, because we know how reactive and modifiable hydroxyl groups are, we can make enormous numbers of carbohydrate derivatives, and we're going to just kind of scratch the surface in this chapter. I just want to make you guys generally aware. You can make sugar acids, sugar alcohols, deoxysugars, sugar esters, amino sugars, and glycosides. There's so many things you can do to monomeric and polymeric carbohydrates in terms of modifying their surface chemistries and fundamentally changing their biological roles. So let's go ahead and we're going to kind of zip through these. So before we do that, one of the questions, because I really like to point out questions that you're going to absolutely see on multiple choice style exams, would include reducing sugars. So what is a reducing sugar? What is not a reducing sugar? A reducing sugar is a sugar which has a free anomeric carbon. So if the anomeric carbon is not bound up as part of a bonding event, if you have a free anomeric carbon, it's going to be reducing because of its unique chemistry. And so these will reduce oxidizing agents such as peroxides, ferrocyanide, and a couple of different metals. And a lot of these result in the formation of sugar to sugar acid. So that's what a reducing sugar is. So let's see here, oxidation of D-glucose to sugar acids. So you can form oxidize D-glucose at a bunch of different positions. You can oxidize it at the 1 carbon, forming a carboxylic acid at the position which used to be a aldehyde, and this would be gluconic acid. You could also then have gluconic acid undergo cyclization of forming a gluconolactone, which totally different structure with the lactone as part of the cyclic structure. You can also oxidize at the C6 position, and you can oxidize at both the C1 and C6 positions. If you oxidize at the C6 position, this is glucuronic acid, and you're going to see this used in modifying the structures of connective tissues quite a bit. So yeah, sugar acids, fairly common modifications. Places you see some of these derivatives, sugar alcohols formed when they encounter mild reductions. Your deoxysugars are going to be really important as part of DNA and RNA. If you have deoxyribonucleic acids, that's because it's a deoxyribose, which is a sugar moiety. You have sugar esters, which are really important in phosphate esters, particularly ATP. I think you guys have probably heard of ATP, so you can see the sugar esters are going to be very important. You have amino sugars, where amino groups replace the hydroxyl groups, and these are going to be really important for connective tissues and cartilage. And then we have glycosides, which are going to be really important for forming oligo and polysaccharides, which are polymeric versions of these monomeric sugars. So not only can you modify these sugars in tons of different ways at the hydroxyl groups, but then you can make polymers of them, which are incredibly varied. And if you just use your imagination quickly, you can just think of the spectacularly complex and varied chemical surfaces that this can form, in addition to sugar being interesting as energy. So sugar alcohols, you've heard of all of these. A lot of them end up in your gum. You can't digest them to get calories. However, the bacteria in your mouth can. So even though you're not getting calories from chewing this gum, the microbiome in your mouth doesn't care. It's going to eat them up and use them as fuel, produce acidic byproducts, and cause cavities just the same. So sugar-free gum doesn't mean that the sugars in your modified sugars in your gum are not being consumed by the microbial organisms in your mouth. But we have our sorbitol, mannitol, xylitol, glycerol, ribitol, and inositol. So lots of sugar alcohols you've probably heard the name of. Deoxy sugars. Deoxy is when you remove one of the hydroxyl groups and replace it with a free proton. You're going to see these deoxyriboses in particular in DNA, deoxyribonucleic acid. You're also going to see it in some unique compounds like rhamnose, which is a component of ubiane, which is a cardiac glycoside. It's particularly toxic cardiac glycoside. Sugar esters. We have our phosphate modified sugars. These are really important for energetics, incredibly important for activating these positions to be superior leaving groups. So when we talk about the bioenergetics of primary metabolism and the rationale for why we would add phosphates to these sugars, we'll get into that next semester in gruesome detail. However, our glucose 1-phosphates and fructose 1,6-bisphosphates especially are important in terms of metabolic compounds as primary metabolism. And our amino sugars, glucosamine and galactosamine. I'm pretty sure everyone here has heard of glucosamine. It's a supplement people take to make their joints feel better. Maybe you don't take them yet, but I'm sure there are 3 people in this class that do at this point. And if you don't, I'm pretty sure your parents have. Because if you take it in very high doses, it's actually pretty effective at helping with joint pain. And glycosides. So glycosides, these are just very simple. Methylglycosides are when the hydroxyl group gets modified by forming a new oxygen-carbon linkage at the anomeric carbon, the C1 carbon. So this does not change the alpha-beta designation of the anomeric carbon. And it doesn't have to be just a methyl group. And because of that, we can form new oxygen-carbon bonds to all kinds of things, including other carbohydrates. And this is called a glycosidic bond. So this is a good way to transition into oligosaccharides and polysaccharides because we are now leaving monosaccharides for the rest of this lecture. And anytime we start linking these carbohydrates monomers together, we're going to add layers and layers and layers of complexity to the structures that we're dealing with. So the most simple variation of non-monomeric carbohydrate is our disaccharide, and that's our simplest oligo. That's when you have 2 monosaccharides that are linked together by a single glycosidic bond, and each of these monomers are commonly referred to as residues. So if you hear in a paper or somebody talking about carbohydrate chemistry and they refer to something as a residue, they're just referring to a monomeric carbohydrate somewhere within the oligo. Key point when we're looking at pictures of disaccharides, if you were asked to pick out which ones are reducing and which ones are non-reducing. If you have a free / unsubstituted anomeric carbon, it's still going to be reducing. However, and this is one of those classic multiple choice questions, sucrose is a non-reducing sugar because both of the anomeric carbons are involved in bonding one another. So let's take a look at that. Sucrose, which we have over here in the bottom left corner, you can see that both the anomeric carbons are binding each other via glycosidic linkage. Therefore, there are no free anomeric carbons, and this is a non-reducing sugar, unlike our free anomeric ends, which we have over here highlighted in green, for lactose, maltose, isomaltose, etc. So trehalose. This is a fun example of a natural protectant produced by insects. I like how they put the quotation marks circulate heolymph, which is, bug blood. So their blood sugar is trehalose, and it's an unusual non-reducing disaccharide. So if you were to be asked, I haven't seen that in a multiple choice quiz before, but it would be another non-reducing sugar. But it likely acts as a cryoprotectant. And we're going to look at a couple more sophisticated cryoprotectant carbohydrates down the road here. But carbohydrates can, because of hydrogen bonding, wrap structurally, because of their flexibility, around nascent or preliminary ice crystals. And this prevents the formation of large ice crystal structures. And the formation of those large crystal structures are really what damage the cellular components during freezing, and are responsible for killing simple organisms when you get large crystalline structures ripping the cell structures apart. And so trehalose is likely a cryoprotectant. We have a couple others here that are higher level oligos that are just examples that the book likes to throw in. And I like these because it introduces the topic carbohydrates having much more sophisticated roles other than just energetics. Here we have erythromycin and streptomycin, which are both really classic broad-spectrum antibiotics. So these exert really potent broad-spectrum antibiotic actions and have been around for a very, very long time. And they're just carbohydrates and modified carbohydrates. So we have a couple different terms. Once again, when we're discussing polysaccharides, a homopolysaccharide is polysaccharide which contains just a single monosaccharide repeating over and over and over and over, whereas heteropolysaccharides have diversities of monomers, monomeric residues throughout the polysaccharide. So some of the classics when we're talking about polysaccharides, and going back, because I know everyone's grade motivated, you want to know what the classics are, you want to know what pops up in questions. These are the classic carbohydrates. Starch and glycogen are your storage carbohydrates. Chitin and cellulose are your classic structural carbohydrates. Those are the four you're really, really going to want to commit to memory going into a multiple choice exam and their various chemical attributes. In addition to starch, glycogen, chitin, and cellulose, we're going to go into recognition, surface recognition, chemistry, and cellular surface modifications of polysaccharides a little bit. The book has a lot of really good examples, and I encourage you to get into the chapter a little bit and read up on those examples. But truthfully, I don't want to add another 30 to 45 minutes to this lecture. It's already going to be a very long lecture. So because of that, enrich the spectrum of examples through some independent reading, please. So starting right off the bat with starch. Starch is plant energy storage. And there's two forms of starch. You have your amylose and your amylopectin. And most starch, this is, broad generalizations. There's always going to be changes to this, varying from organism to organism. But most starch is 10 to 30% amylose and 70 to 90% amylopectin. And the branches occur in the amylopectin. And branches in amylopectin in starch occur every 12 to 30 residues. So we have our amylose, which are nice straight chains here. And we have an amylopectin, where we have a branch where the anomeric carbon binds to 6 carbon at the branching position. So, excuse me. This is your 1,6 linkage. So your classic amylose is your 1,4 linkage, where you have your anomeric carbon here, your 1, and then we go 1, 2, 3, 4 around the ring. Well, 1, 2, 3, 4, that's your 1,4 linkage. And you are always going to have a reducing end free. Here we have a 1,6 linkage, and because this is a now bound anomeric carbon, and we don't have a free anomeric on the opposite side of the 4 position here, all of these are bound. This branch is non-reducing, even though we have reducing ends on the very end of the amylopectin, where I'm circling in the bottom right-hand corner. Now, if you do that iodine starch test, the reason you see a formation of purple is because iodine neatly fits right into these helices, which form when amylose is in aqueous suspension, and it gives you that really dark blue or purple characteristic color for your iodine test. Just fun mechanistic insight into the stuff we did in 100 level. So why is there branching in starch? Let's go back real fast. Why on earth would we have all this branching? Give everyone a second to think about that. Usually this is where I would start asking people questions in class, and either I'd find out who likes to wrap their head around problems and throw some spitball answers at me, or who read the text ahead of time. And at this point, if you read the text, you already know, likely. The chemical rationale is because phosphorylase enzymes, which release the glucose 1-phosphate product from amylose and amylopectin chains, need to access the ends of the chains. And the more branching there is, the more ends of chains are available for the phosphorylase enzyme to access and attack. So the branching is a mechanism by which this large polymeric compound can be sequentially deconstructed to form glucose 1-phosphate as a product. So it can be liberated and then moved into primary metabolism for the plant. Now, what's glycogen? We just talked about starch. Now we're going to move into glycogen. Starch is carbohydrate storage for plants. Glycogen is carbohydrate storage in animals. And let me move back real fast. Note that all of these carbohydrate residues are beta-D glucose because of its structural characteristics. And here we have a branching in glycogen. So glycogen is how we store polymeric glucose in animals, in ourselves. And we carry around quite a bit of it at any one time. As much as 2% of our muscle tissues mass can be glycogen when we're all carb loaded, and the liver is the same way. About 10% of the liver can be glycogen. Now, whenever you are doing one of those low carbohydrate diets or fasting diets, people go, oh man, it's so fantastic. I lost 5 pounds in the first 3 days. Yeah, you lost all of your glycogen because you are on a very low carbohydrate diet and you went through all of your reserves of muscle and liver glycogen. That's why you're feeling a little bit logy, a little bit, not functioning super fast yet because you haven't kicked over to ketogenesis. Your body thinks it's starving, but hasn't quite kicked into its emergency backup modes. And what you've done is you've kicked out a whole bunch of weight of glycogen, and also glycogen increases a little bit of water retention in that tissue. And with the loss of glycogen, you lose a little bit of water retention. So you've lost some water weight as well. You haven't actually burned any fat, or you've burned trivial amounts of fat. So don't be so encouraged by that initial 3 to 4 day weight loss on those low carbohydrate. You're just getting rid of some glycogen. The only difference structurally between glycogen and starch, and amylopectin specifically, is that it's even more highly branched. So the alpha-1,6 branching, instead of occurring every 12 to 30 residues, happens every 8 to 12 residues. And the reason for that is because as animals, we need to liberate that glucose even faster. So in glycogen, it's even more branched. And if you were to put iodine in solution of glycogen, you would get the exact same violet color as a positive test, even though it wasn't starch, because of its structural similarity. Dextrans, I'm just going to throw a little bit of curveball in here. Dextrans are another class of carbohydrate which have a slight but very important difference from starch and glycogen. If you change the main linkages between glucose from alpha-1,4 to alpha-1,6, you get a whole new family of polysaccharides, and these are considered dextrans. And you can be branching, do 1,2 branches, 1,3 branches, 1,4 branches, and these are really commonly formed by bacteria and are a major component of our dental plaques. We don't make a lot of dextrans, but other lower level organisms do. They're also super useful in certain techniques in biochemistry, which help us do column chromatography. One of the media we use called Cefidex is a dextran-based component. So just that's a little bit of trivia. So moving on from our energy storage polysaccharides, let's talk about our structural polysaccharides. There's a couple notable differences. Starch and glycogen linkages primarily consist of alpha-1,4 linkages, and this allows for the curvature to the structure of the carbohydrate. So let me see if I can zip back. Here we go. This curved structure is because of the type of linkage between anomeric carbons. We have an alpha-1,4 linkage, which gives the kink to the structure. Cellulose is a beta-1,4 linkage, and this creates long linear structures. And this long linear structure provides a lot of physical and structural strength as part of a polymer. So to review, cellulose is your classic structural polysaccharide. It's the most naturally abundant polymer on Earth because it's ubiquitous to all plant structures, all trees, all hemps, all bamboos, all gain strength from your cellulose structure. It's found in nearly every single cell wall of nearly all plants. Once again, there's occasional exceptions. It's insoluble. So whereas starches are soluble, which is important, this structure is insoluble and highly organized and helps, which makes sense because even though it's a carbohydrate, we don't want it to melt whenever it rains in the forest. Fun fact, I've been listing all these trees. Cotton is nearly pure cellulose. So here's a visualization of the difference between your alpha-1,4 linked beta-D-glucose, where you have a curvature with the alpha-1,4, and then because you have everything flipped alternately, the beta-1,4 is nice and linear. You can extend it, nice linear extended conformations. And in addition to having nice long linear structures, because of the organization of all of the peripheral hydroxyl groups, we can get extensive planar and interplanar hydrogen bonding, which makes for very, very strong structures. A couple other classic structural carbohydrates, not as important as cellulose, but, well, unless you're a bug. You have chitin. This is really important as a structural compound in exoskeletons of crustaceans, insects, spiders, and in cell walls of a couple different types of fungi. And it's really similar to cellulose, but all the second carbons are acylated, nitrogen acylated. So, slight modification to the monomer, but the polymer is fundamentally the same. And we are going to skip some of these. However, glycosaminoglycans, we will get into in greater detail in a little bit. Let's see here. I probably could have pulled a few of these things out. I'm not going to talk to you guys about agarose. Agarose is used in the laboratory as a gel. High levels of agarose make the migration of biological molecules through a gel that has been exposed to an electromagnetic field more difficult, but I'm not going to go into significant detail on agarose here. Here we go, glycosaminoglycans. So glycosaminoglycans are linear chains of repeating disaccharides where one unit is an amino sugar and the other and one or the both are negatively charged. And so these would be highly charged polymeric compounds. And a few of these you may recognize. We've already talked about heparin a little bit. There's also chondroitin. There's hyaluronate, which I always struggle to pronounce. There's two different variations of chondroitin. There's the 4-sulfate and 6-sulfate. There's keratan sulfate as well. And chondroitin and hyaluronate or hyaluronic acid are also really important compounds in the structure of your cartilage and connective tissues. So what are some of these functions other than connective tissues? So heparin, we already talked about that in one of our case studies. It's a natural anticoagulant. The hyaluronates can be very large. They can have 25,000 different disaccharide units, so up to 50,000 monosaccharide units. And they're in synovial fluid. They can have some function in the eye, and they help with joint lubrication in general. Chondroitins and keratan are involved in tendon, cartilage, and other connective tissue structures. Dermatan, which I skipped over in the previous slide, is in the skin. And all of these glycosaminoglycans are also constituents of a type of modified protein called the proteoglycan, which we'll get to at the very, very, very end of the chapter. Now, many of you have had some basic microbiology, and I'm not going to go into significant detail on the differences between gram-positive and gram-negative bacteria and the chemical constituents of their shells which make up these differences. I'm just going to skim through here, so I'm going to be zipping through some slides because I don't want to take this out way past an hour and 15 minutes for everybody's well-being. Gram-positive bacteria have a single phospholipid bilayer and a thick peptidoglycan outer shell. Gram-negative have two membrane phospholipid bilayers with a thin peptidoglycan shell between those two bilayers. And what this looks like structurally, when we're talking about, let me zoom out a little bit. This is more detail than I'm hoping to introduce. Well, actually, for those of you who are interested, the difference between your gram-negative and gram-positive is here in the modification of that lysine. But when you zoom out, you get a bigger difference. So here we have a gram-positive, here we have a gram-negative. So gram-positive, you have your pentaglycine cross-link between your alanine, glutamine, lysine, alanine subchains. And here we have the exact same chains in the gram-negative, but you have a direct cross-link, which is a lot tighter. So you got the same tetrapeptide units, alanine, glutamine, lysine, alanine, but you don't have that long crosslinker in your gram-negative. Here we go. So here we have our gram-negative. Zoomed way out, you get these long lipopolysaccharide hairs that come off of your two layers. So you've got our outer lipid bilayer, you've got your peptidic glycan in the middle, which is tightly cross-linked, and then you've got your inner peptidic glycan, and then you have these long polysaccharide chains. And the LPS, or lipopolysaccharide, is commonly used to initiate inflammatory pathways in controlled immunological experiments because all of your immune cells recognize LPS, lipopolysaccharide, and will amp up immune response aggressively when exposed to this particular carbohydrate polymer. Now if we go, back to our gram-positive, you have that long cross-link between our tetrapeptide, and you have your single bilayer followed by this thicker shell of peptidic glycan in your gram-positive. There we go. So what's the structure of lipopolysaccharide in your gram-negatives? It's these long hairs of carbohydrates that are just drifting out there. And this is a really neat way to introduce the roles of carbohydrates on the surfaces of cells. So not only do bacteria have carbohydrates on the surface of their cell walls, so do we. And that's because they make excellent sensors, and they have fantastic mobility, and they have the ability to be highly varied in terms of their chemical surface chemistry. Because of that, we see a huge diversity of glycoprotein and proteoglycans, and we're not even going to scratch the surface in this. This is about becoming aware of some of these topics, research topics. But they're particularly important for regulating cell-to-cell recognition and interaction. And once again, a classic case of this is immune response. Macrophages, for instance, will proliferate due to a number of different interactions with their environment. But one of those interactions is cell-to-cell contact. If you get too many macrophages within a certain density, they recognize their own presence and think of this as a immune insult and will activate even more due to their density. So in this particular case, these cells can recognize each other and proliferate aggressively. Other cells, like neurons, when they come up close to one another, will stop differentiating and stop proliferating and will slow down because of that cell-to-cell contact. So based on the different biological roles that cells have in terms of the organism, different regulatory events can take place just via cell-to-cell contact. And most of these interactions, regulations, are going to take place through glycoproteins and proteoglycans. So what are glycoproteins? How do they function in cells? Well, they can be nitrogen-linked or oxygen-linked, which gives them a huge amount of diversity structurally. The nitrogen-linked saccharides can attach via the amide nitrogens of asparagine residues specifically, and the oxygen-linked saccharides attach to hydroxyl groups of serine, threonine, and hydroxylysine. So here we have oxygen-linked saccharides attaching to serine. Here we've got oxygen-linked saccharides attaching to threonine and serine. And here we have nitrogen-linked saccharides attaching to asparagine. And you can see, whereas the first ones were just a couple really simple, here we have some very complex modified. We've got some nice 1,4 beta-1,4 linkages going on here. Then we've got our alpha-1,6 linkage. We have a couple mannoses out here. We've got some modified carbohydrates going on with nitrogen modifications. And zoomed out even more, it could get progressively even more complex. So some of these are very high in mannose. That's one of their classic characteristics, which we see particularly in the high mannose over on the left. And then we have some really complex variations on end-linked saccharides shown over on the right side. So the O-linked saccharides often form extended bristle brush structures, which function as, for the lack of a better word, I'm going to say sensor, or functional domains, which on the outside of the glycocalyx, which is the outside of the cell wall, which has been modified by your carbohydrates. And this is just a simple variation on this, where you have these long bristle brush structures that reach out from the cell wall, and they can be unmodified like we see over here on the left, but they can also have sensor sections like LDL receptors over here on the right, which get you a receptor that is well spaced off of the cell wall, so it can kind of fish around for a little LDL in this particular example, and then that, when bound, can communicate a signal down through the wall of the cell, and we can get transmission of information from outside of the cell to inside of the cell, so the cell can start to respond via contact with some analyte in the system around it. I love this title. Functions of nitrogen-linked oligosaccharides. Many functions are known or suspected. And that's the truth. It's incredibly diverse. Oligos can alter the chemical and physical characteristics of proteins dramatically. It can target them for packaging, for transport, for subsequent modification. It can protect against proteolysis. It can be indicative of the age of a protein as the saccharides are subsequently slowly degraded, and then once they're shortened to a certain critical length, can be targeting those proteins for being taken out of circulation and replaced with new proteins. It's just a fantastically diverse set of modifications. Oh, here we go. Antifreeze functions of glycoproteins. So, whereas before we had talked about the formation of hydrogen bonds with these carbohydrates around nascently forming ice crystals as a mechanism for antifreeze protection, there's a couple of different classes of glycoproteins which have repeating units of A, and T, where we can see multiple repeats, as many as 50 units of this, and you got alanine, threonine, and the threonine residue specifically is glycosylated with a disaccharide. So you get these long flexible rods with regions of threefold left-handed helices, and they can wrap around ice crystals very effectively. So this is what it looks like. You've got this alanine, threonine, and on each threonine you have this residue, dimeric residue of carbohydrate coming off of it. And each of these dimers can then wrap around nascent ice crystals and prevent aggregated larger crystals from forming, which can prevent or slow the damage to cell walls and cellular structures. I was talking about timing devices. This is a sialic acid residue, which let's see here, we start up at the top. If all these sialic acids are present, then this isn't bound by the liver for degradation, but as this goes through circulation, the sialic residues occasionally get bumped off, exposing the galactose. This is still poorly bound in the liver, but then you get moderate binding once it gets even aged, and once all the sialic acids have been kicked off, this is tightly bound by the liver and targeted for degradation because it's time to remake some new protein that hasn't been working for too long. I think this is a little bit redundant. Proteoglycans, okay. And we're entering some regions of examples in the book. I probably could have edited out these last slides. Functions of proteoglycans. We've talked a little bit about how binding of growth factors can be accomplished at the surface of the glycocalyx, and also how they're involved in cushioning of joints and retention of water in the joint, which gives you shock absorption. And good, that is. And so this was an enormous amount of information. We got pretty well into the stereochemistry of monomers. And then we talked a little bit about the classic polymers, both the structural and energetic storage polymers. And then we went down, we didn't even go down any rabbit holes when it came to proteoglycans or the diversity of ways that carbohydrates can function biologically in addition to being energy or being structural. There's really just, we could go on for, we could write books and books and books on the topic. So when we get into the take-home type questions on this, you may have to do a little bit of digging because there's a good likelihood you'll have to do a little bit of digging. But there's a lot of great information out there. And if you're concerned about multiple choice type questions, keep in mind those things that I talked about earlier in the lecture. So this is going to be it for carbohydrates and proteoglycans. And I'll be talking to you guys later.